If you watch enough science fiction movies, you likely have a very specific image of an acid in your head. It appears as a glowing green liquid capable of melting through industrial metal decking or a bank vault in seconds. Yet the reality is much more domestic. You encounter acids every single day in the tea you brew. Simplest atom in the universe, hydrogen. It consists of exactly one negatively charged electron orbiting a single positively charged proton. Chemically, an acid is any molecule willing to give its hydrogen atom away. However, as it lets go, the acid molecule keeps the electron for itself, forcing the hydrogen to leave its only negative charge behind. This leftover hydrogen core is now an ion. An ion is an atom that carries an electrical charge because it has either gained or lost electrons. Without its negative electron, this hydrogen ion is simply a solitary, positively charged proton. These naked protons are highly reactive. Because they carry a positive charge, they aggressively seek out new electrons from any nearby structure to reach a stable, neutral state. When an acid eats through a material, it is actually performing a violent chemical theft. The acid's protons are ripping electrons directly out of the surface they touch, pulling the solid material apart at the molecular level. Svante Arrhenius developed the first clear definition for this behavior in the late 1880s. He discovered that these molecules release their hydrogen protons specifically when they are dissolved in water. When surrounded by water, the hydrogen proton finds neighboring water molecules more attractive and jumps ship. It attaches to the H2O to form a hydronium ion. A hydronium ion is a water molecule that has gained an extra positively charged proton. Every acid follows this pattern, but they vary in how effectively they release those protons. We measure the strength of an acid by the fraction of its molecules that actually drop their protons into the water. A strong acid, like the hydrochloric acid in your stomach, has very little grip on its protons. When dissolved, almost every molecule in the solution releases its protons simultaneously, flooding the liquid with reactive ions. In contrast, acetic acid, the acid in vinegar, is a weak acid. When it meets water, only a tiny percentage of the molecules release their protons, while the vast majority remain bonded. The chemical reaction itself remains identical across every acid. The perceived power of the liquid comes down to the density of free reactive protons floating in the water. In 1909, a Danish chemist named Søren Sørensen developed the pH scale to measure the concentration of these protons. The letters pH stand for power of hydrogen, providing a standard way to quantify acidity. This diagram shows the pH scale, running from 0 to 14. 7 represents neutral water, while the range from 0 to 6 marks increasing acidity. This logarithmic structure means that dropping from a pH of 3 to 2 represents a 10-fold increase in the concentration of hydrogen ions. This scale allows us to measure everything from the stomach acid that aids digestion to the energy-producing ATP in our cells. ATP, or adenosine triphosphate, is a biological molecule that releases hydrogen ions to provide energy for your body's functions. Acids rely on a chemical counterbalance called bases. If acids are proton donors, bases are proton acceptors, molecules that actively seek to take in the protons that acids give away. When an acid and a base meet, they trigger a neutralization reaction. Neutralization is a chemical process where an acid and a base interact to reach a stable, neutral state, effectively canceling out each other's reactivity. This exchange results in a peaceful chemical resolution. Once the protons are accepted by the base, the corrosive properties vanish, leaving behind only harmless water and salt. These reactions are essential to human biology. Amino acids use this proton-sharing ability to link together and build proteins, while nucleic acids use similar properties to store and protect your genetic information. These molecules function as generous donors, facilitating the chemical exchange necessary to digest food, store genetic data, and maintain the biological balance of our world.